Well, earlier today, we had a really nice discussion of inflammation. So now we're very lucky because Jess Gregory is going to come up and focus on metabolic turnover, inflammation, and redistribution.
All right, thank you. The last few speakers have really nailed in about specific nutrient requirements or directions that the field needs to go. Mine is much different. I'll talk about what's unknown, what are research priorities, and I'm going to talk about vitamin B6 as an example, which I just realized this is my 43rd year in the field of B6 and other B vitamins, so kind of scary. So up until the last few years, we thought we knew a lot about the nutrient requirements for this vitamin in humans and with good rodent models or other animal models. That's changed considerably, so let's proceed. So the vitamin B6 status is generally adequate in most developed countries. Combination of foods provide an adequate amount of the vitamin, and with relatively few exceptions based on extremes of food selection. Healthy individuals get their B6 from regular foods plus dietary supplements, and that's certainly adequate for the most of the population. So I should say that the cutoff for the primary biomarker used in screening studies is pyridoxal phosphate, and as of the 1998 DRI study, that was defined as 20 nanomoles per liter of circulating pyridoxal phosphate, or PLP. The most recent published N. Haynes study that showed B6 data from my friend Christine Pfeiffer showed that about 10 to 12% of the N. Haynes population of all the different groups was on the less than the 20 nanomole per liter. So I think I would conclude that most of the population's in pretty good shape. But we don't know why that 10% or so is showing insufficient PLP biomarker. I mentioned that the RDA is based on this cutoff. Sorry to repeat. We've done a large screening study looking at women consuming oral contraceptives in Gainesville, Florida. They're all very healthy and under 30 nanomoles per liter, which I would consider marginal. Only 13% showed that, so we're in reasonable shape, but it's not perfect, as we'll see. So the most common biomarker is plasma PLP. It's easy to measure with modern analytical equipment. It can be automated in high throughput, and it generally parallels intake, and 25 years ago, it was shown that it parallels liver PLP content. And so that's really the physiological value of it. When that's pyridoxal phosphate, I won't wade through all this, but this is a summary of vitamin B6 metabolism. These are the absorbed forms. And later on, I will discuss a bit pyridoxic acid. That is the excretory form detected in plasma before it's excreted in the urine. So that's also not a bad biomarker, but again, there's some problems that we'll see. So pyridoxal phosphate, the last count was over 160 enzymatic reactions in the body, and these include a lot of different amino acid metabolism. gluconeogenesis from gluconeogenic amino acids, glycogenolysis, and a lot of one-carbon metabolism where there are 4 or so PLP-dependent enzymes, and tryptophan metabolism. We'll discuss that later. Sphingolipid metabolism, and we showed some fatty acid influences in B6 deficiency. And the thing in the field that still exists is a real uncertainty in what's the sequence of what goes wrong in insufficiency in the people that are in low B6. You can see there's a lot of metabolism that involves pyridoxal phosphate. So disease relationships since the mid to late 90s, there have been a number of papers that have been published that show a relationship between risk of vascular disease, which includes heart disease, risk of death from heart disease, stroke, venous thrombosis, all of which have vascular common denominators. And in these epidemiological studies, if you're either under 20 nanomoles per liter PLP or under 30 in some, your risk goes up. Low B6 intake in another study was shown to be a cardiovascular risk factor in the Nurses' Health study. C-reactive protein, which is kind of a common inflammatory marker, measured in epidemiological studies of vascular disease, tends to, for elevated C-reactive protein, PLP tends to be lower, so there's this inverse relationship. And in inflammatory bowel disease and rheumatoid arthritis, both inflammatory diseases, the disease severity is inversely associated with plasma PLP. So the worse the inflammation, the lower the PLP, but in the red cells, erythrocyte PLP stays normal. So there's something different in the pools and their mobility. So what I want to show is a series of papers and studies, some of which ours, some belong to 2 dear friends of mine, Jakob Selab, this one, and others from Per Ueland and University of Bergen, Norway. So here's a Selab study that they observed that PLP was inversely associated with many biomarkers, not just CRP, is inflammation. So this is a subset of N. Haynes, I believe. They've also done the same thing in Framingham. And so this doesn't show real well, but that is an aggregate of a bunch of inflammatory markers. And here is, plasma PLP. So people with high B6 intake, it's up here, more inflammation, their plasma PLP goes down. Same thing with middle, same thing with low. So what? Well, plasma PLP in inflamed people is not a good biomarker is one conclusion. And second of all, that if you're looking at B6 intake and the percentage of the people that were less than the PLP, this is normal for low inflammation. High inflammation, you see this sort of thing with relatively, this is kind of normal B6 intake, and a high percentage were showing insufficiency. I think the percentages may be a bit skewed by the assay they used, but still the trend holds that inflammation certainly influences the incidence of low PLP. So why is that? A couple of hypotheses, one of which is that pyridoxal phosphate can be redistributed from the more kinetically mobile pools to sites of inflammation. And why would that be? Well, all of these different metabolic processes, the sphingolipid, tryptophan catabolism, a lot of one-carbon metabolism is necessary for cell synthesis and replication, transsulfuration to make sulfur amino acids, hydrogen sulfide, and glutathione, and a bunch of other things. So that's one hypothesis with some evidence. Second is that is there some acceleration of B6 catabolism? And if you measure the amount of pyridoxic acid peed out in a day's time, there's not a lot of change. However, it has been shown by my Norwegian friends that, where is it here, if you take the ratio of pyridoxic acid in plasma and divide it by the two measurable active forms, PLP and pyridoxal, that ratio is increased, suggesting, suggesting that there is enhanced catabolic process going on along with inflammation. So we don't need to dwell on this, but the compartmental modeling of B6 has been very thorough, and basically what they show was 70 or 80% is locked in muscle to glycogen phosphorylase and not mobile and not responsive and slow turnover. However, the other 20 to 30% is in liver and all the other organs, so some of those are quickly kinetically active and others pretty much stay put, and those are the sites that can be depleted if there's a demand for inflammation in some other tissue or some other organ. So that's what a research need is really to define what happens from the original sites as there might be some relocation. So what's the evidence for relocation? Another Celeb study. They use the rat model of arthritis, and here is main thing I want to show here. This is liver PLP before, before the control and after the adjuvant injection. This is, I think, 2 weeks later and 4 weeks later, and you see the persistent difference. In other words, the liver's given up some PLP to go somewhere, presumably to support the inflammation. Plasma is also the same thing. That's reduced. Muscle is not. So if PLP has its limitations for inflamed conditions, inflammatory conditions, what might be better? Well, the old traditional erythrocyte transaminase stimulation assay, which is a very large pain to run on a large scale, erythrocyte PLP, which might be very good, but it's, again, difficult to extract PLP efficiently from erythrocytes, and it's not well standardized. Urinary pyridoxic acid requires collecting urine, and there's cycling of that, and probably the most effective metabolic marker in the things that we have studied is plasma cystathionine. So cystathionine is an intermediate in the transsulfuration pathway that takes homocysteine to cysteine and then glutathione. In B6 insufficiency, cystathionine goes up, but also in B12 or folate deficiency. So if you rule out the other two, it's a good B6 marker. The kynurenines have been known for many years to be altered in B6 deficiency, and so now that we have really sound analytical methods for studying by LCMS the array in the tryptophan pathway, then we can analyze that. So I'm going to skip that in the interest of time. That's a rat study that's sort of perplexing. So the tryptophan catabolism system pathways is this. There's constitutive and inducible, so inflammation can induce the alternate oxidase, indoleamine dioxygenase, and the first product is kynurenine. Two different B6 dependent pathways can take that to kynureninic acid and anthranilic acid, hydroxykynurenine. Same two kinds of enzymes can produce xanthorenic acid and hydroxyanthranilic acid. So if you take any one compound that's influenced by inflammation, because the pathway is influenced by inflammation. However, if you take the ratio to indicate substrate and product, that reflects B6 status of the relevant enzyme. And so it's then possible to plot in healthy people PLP versus this ratio, hydroxykynurenine to xanthorenic acid, and that looks like a pretty good biomarker that is very little influenced by inflammation. Same thing, you can make other ratios, and those turn out to be very effective biomarkers. They haven't been well validated, but they're looks like the wave of the future in B6 assessment from a functional biomarker. So we've done essentially the same thing that validates that, and in healthy people, the ratio in B6 adequate and B6 restricted people down to a level that's near the cutoff. There's the difference in PLP, there's the difference in the ratio. Its slope's a little different, but does look encouraging. Okay, in collaboration with the Ullin group, they had made the observation that in cardiovascular patients, they have determined that the elevation of the intermediate cystathionine that I mentioned if that's elevated in these heart patients, that's thanks, Steve. So anyway, cystathionine is a risk marker for both cardiovascular disease and death from cardiovascular disease in their studies. So we conducted metabolomic analysis on that, and I won't deal with all the observations, but with respect to B6, we found that the low cystathionine had pretty much adequate pyridoxal phosphate, the high cystathionine, low PLP. So we then look at the ratio, and that's pretty much the same. So this is an example of the inflammation in this population, making the PLP biomarker unreliable, whereas the ratio is the effective thing. Within the metabolomic analysis, the orange are the biomarkers that differentiate the high versus low cystathionine groups, and what we see are a bunch of tryptophan metabolites in the one end and kidney function in the other. So there's more than metabolism going on, but there is certainly some evidence of a lot of different alterations in this population. So in summary, PLP is a very good biomarker for most of us, which are well nourished people. And the questions raised by its responsiveness to inflammation certainly raise questions about its value in population evaluations, such as in Haynes and such as developing an RDA for B6, particularly in populations that can be subject to inflammation, many kinds of inflammatory disease. The ratios of tryptophan catabolites do seem like an encouraging approach to developing a biomarker that is less sensitive to inflammation, but we need the validation in terms of cutoffs and adequacy and deficiency and so on. Some of the data that we've generated, and certainly some of the data from Norway, will be useful in making those assessments. So we don't know about why inflammation triggers a redistribution of B6. There are plausible mechanisms, as I discussed, and those are certainly research priorities. Other B vitamins, we don't know about the same sort of thing, but the same kind of needs for metabolism in inflammatory conditions would certainly apply. So thank you for your attention, and I'll be glad to answer questions.